Hi, I'm Vasilis and I'm going to talk through the first section of the Data Management Plan, DMP, describing your data. By the end of this video, you will be able to understand and fill in the first section of your DMP application. Remember here that these are just suggestions of what I would write for a small gene expression study to give you an idea of granularity of content and what I would consider writing. So, previously I showed 9 sections that I'll consider in this video series, noting that not all funders ask for these to be written. I'm starting here with the first of these. Let's start with the section number 1, the description of the data. This section consists of 4 subsections. The type of the study, in which we should provide some general information about our experiment. So, view this as an executive overview of activities in the grant that will generate data. Here I'm working with human data, so I would like to reference this specifically because it is relevant to later sections around data security and sharing, since human data can be subject to control or closed access. In our example, we have a transcriptome analysis of diabetes type 1. It is an RNA-seq experiment using human tissue from consenting donors. The size of the experiment is 12 participants for the control group and 12 participants for the disease status. However, I'm also looking to complement my findings with gene expression data in the public domain. Subsequently, we need to provide information about the type of the data that will be generated from our experiment. Again, this is an overview before we talk about specific details such as file sizes and file types. They are quantitative measurements of gene transcript expression in human tissue using bulk RNA-seq. In case we are using publicly available data of the same or even another organism, we should also mention it. In this example, we will also use mouse gene transcript expression data from publicly available microarray and bulk RNA-seq studies. Next, we need to provide information about the origin of the data. The data for the human samples will be generated as part of the study, so they are not currently available in any public domain, but the mouse data that will be used are publicly available in GEO. Here I wanted to stress that the data I propose generating isn't already in the public domain. And finally, we need to provide information about the format and the size of our data. In this section, we need to provide as many details as possible, and the funder templates usually list the sorts of information you can provide. Here I wanted to state the instrumentation generating the data, as well as data sizes and file formats. By stating file sizes, I'm able to estimate and resource storage requirements. By stating file types, I'm able to demonstrate I'm working with interoperable file formats. For human data, we are planning to use the Illumina HiSeq 2500 platform with 100 base pair paired end reads in a sequencing depth of around 45 M. Data format will be compressed FASTQ, and the total file size of the 24 samples, 12 control samples and 12 disease samples, is estimated to be 150 gigabytes. For the mouse transcriptomics data, as this is publicly available data, it will not be archived by this study. The data has been generated by both expression microarray and RNA-seq technologies, and they are stored as compressed FASTQ, cell, and text. The total amount of samples is estimated to be 50, with a total file size of about 100 to 400 gigabytes. Note here that estimates are fine. Remember, the DMP is a working document and numbers and sizes can be amended. Estimates are better than not stating them and often do not change too much. So, that was the second video of our RDMbites series of Things to consider when writing a DMP for a grant. Please see the links mentioned as well as links to additional resources alongside the video.